Hello, everyone. My name is Dr. Jay, and we are back after a pretty long sabbatical. And I hope everyone's doing well. The last videos which I recorded were those of first and second year subjects for those who are exam going and from the university point of view. However, this time I thought that we'll take it up a notch above. And since many of you had requested that a similar series for competitive exam preparation would help, I thought we would restart this series. And since I currently work as a tutor in the Department of Physiology at a prominent Mumbai Medical College, I thought physiology would be the best bet to start with. Okay. So I've collated questions from platforms which provide these. I've modified them a bit and I'll discuss them. What makes the difference is this, the audiovisual connect which we have. Okay. So physiology generally starts with the first chapter, which is homeostasis and cell biology. So without further ado, let's get going. Okay. So what are the objectives which we have? The first one being what exactly is homeostasis, then the feedback mechanisms, and the cellular organelles, including cell membrane, Golgi apparatus, endoplasmic reticulum, mitochondria, and everything, cellular proteins, and clinical questions, questions which have been relevant from the clinical perspective and which are generally heavily being tested upon. Okay. So let's start with the first question. The first question says that homeostasis is known as dynamic equilibrium because all variables are given equal importance. It can achieve a constancy in extreme environmental challenge. The set point for all variables is fixed, or a body maintains an internal balance within fluctuating limits. So what is the answer? Yes, so the answer is option D. Why so? Because let's see. So the term homeostasis was first given by Walter Cannon, right? And a term which predated it was milieu interior, right? And that was given by a French physiologist whose name was Claude Bernard. So homeostasis basically means a constancy in the internal environment despite changes in the external environment, right? So maintenance of a constant internal condition despite changes in the external environment. That's what allows living beings to, flourish despite changes in the environment. So that is homeostasis. And Claude Bernard coined the term milieu interior, which was later redefined or refined by Walter Cannon to term it as homeostasis. Okay. Why not the other points? Because let's see them one by one. The set point for all homeostatic variables is always fixed. So this is wrong. The set points are variable. For example, I'll give you an example of baroreceptor reflex. It operates in the range of 60 to 160 mmHg. So we don't have a single set point. There's always a range of values. For example, if we say heart rate, what's the normal heart rate? It is 60 to 100 beats per minute. What is the normal respiratory rate? It's 12 to 18 cycles per minute, right? So we don't have a set point. We have a range of values over which that constancy is being maintained, okay? Can achieve constancy in extreme environmental challenge. Here, the term extreme is a bit extreme. Extreme environmental challenge, the body or the systems generally cannot cope up with, and that leads to disease. So what is disease? Disease is basically dysfunction, right? Dysfunction leading to loss of regulatory capacities, right? So that is disease. All variables are given equal importance. Again, that's wrong because some variables are given more importance than the rest. For example, let's take the example of, for example, increase in H plus in the blood, which is known as acidosis, right? Now you need to remove this hydrogen ions. How will you remove them? Either you can have a respiratory or you can have a renal compensation, right? So for example, respiratory, I'll give you an example. Increase H plus, that will combine with bicarbonate, which will form H2CO3, which reversibly dissociates to form H2O and CO2. Now the excess CO2, right, the CO2 which is formed is eliminated via hyperventilation. So now the H plus is getting consumed, right? So hyperventilation leads to an increase in respiratory rate. Normal I just mentioned was 12 to 18, but you are having an increased respiratory rate in order to maintain the concentration of H plus within the normal limits, right? So not all variables are given equal importance. Some are more important than the rest. So this question is done. We coined the terms, why the other questions, why the other options are wrong and why option D is correct. Okay. So let's move to the next question. And the next question is, which of the following is not a component of the homeostatic control system, right? So what is the answer? The answer again is D. There's nothing called as an effector, right? So what are the components of a homeostatic mechanism? For example, you have the environment, right? So if there's any change, that is detected by a receptor. So the receptor detects the change. Now that receptor via an afferent pathway, afferent, reaches the integrating center, which generally is the central nervous system. In some cases, in some cases, it can be brain or the spinal cord. Depends. Yeah, as we go ahead, we'll discuss those examples. We have an integrating center, which gives the command, which processes information and then gives commands. Those commands are relayed via an efferent pathway. And from the efferent pathway, it reaches the effector, right? So after reaching the effector, there's a response. Okay. So what are the components of a homeostatic control of a homeostatic control system? It is a change. You have a receptor which senses it. You have an integrating center. You have an effector which is responsible for responding to it, right? And you have a response. So the answer is effector, right? The next question, feedback regulation is seen. Now, for all homeostatic mechanisms, generally you have three types of control systems. Which are the three types of control systems? This question talks about control systems, okay? So let's look at it. Feedback regulation is seen in increased heart rate that occurs in an athlete just before a sprint begins, no. Smell of food triggering secretion of salivary and gastric juices, no. An increase in insulin before glucose absorption has raised blood glucose levels, no. Decrease in LH in response to testosterone, yes. This looks like a feedback mechanism. So which are the three control systems? I'm sure most of you would be aware. We have a negative feedback. We have something called as a positive feedback. And we have a feed forward, right? So how do we define each of them? So negative feedback consists of changes which brings a variable which is either excess or deficient to its normal mean value, right? So you had a variable which has now gone up or which has now gone down. it needs to come back to its normal mean value. That is negative feedback. So majority of your hormonal control systems are under negative feedback mechanisms. Okay. So, and maybe, like LH stimulates the production of testosterone from the Leydig cells, but testosterone in turn inhibits the secretion of LH and GnRH both, right? So that's an example of negative feedback. Rest, all the three options are an example of feed forward mechanism. Now, what is feed forward? Feed forward is also known as anticipatory control, right? So, in anticipation of a change, the body responds first. The change has not yet occurred, but in anticipation that occurs. For example, it says that smell of food triggers secretion of salivary and gastric juices. Now, for anyone who knows the phases of gastric acid secretion, you have a cephalic phase, you have a gastric phase, you have a intestinal phase. So cephalic phase is only the sight or the smell of food. Smell of food triggers secretion of gastric juices. So cephalic phase in gastric acid secretion is an example of feed-forward mechanism. Increased heart rate that occurs in an athlete just before sprint begins. So the body is anticipating that increased workload would be there on muscles and the other systems. You need more of oxygen, you need more of blood to reach the body. So that's again anticipation. Increase in insulin before glucose absorption has taken place. Again, in anticipation of rise in blood glucose level, you have insulin secretion. So all of them are examples of feed-forward mechanism, which are examples of positive control systems. So in positive control systems, you have positive feedback mechanisms. You have something which is very popularly remembered as CLAPS. So C is the clotting, L is the LH surge, then you have action potential, then you have birth, which is parturition, and you have S. What does S stand for? S stands for shock. But we'll see what kind of shock in the next question. Okay. So the next question is, positive feedback is seen in all except. So I just mentioned in the previous question, it is yes, action potential, gastric acid secretion, clotting. Now, some of you might be confused, as to, for example, gastric acid secretion. So the correct answer is gastric secretion. Why? Because if there is shock, what does shock generally mean? Shock means when the perfusion is not able to match the demands of the body, that is shock. And most commonly we talk of hypovolemic shock. So it's said that if we have 5 liters of blood, up to 2.5 liters or even 3 liters loss, our body can tolerate. But if there's more than 3 liters loss, the compensatory mechanisms, which we'll study subsequently, the compensatory mechanisms fail to bring back the perfusion to the normal levels. And as a result, there's widespread injury, the blood pressure progressively decreases. So that is decompensated shock. So decompensation means it is not compensated by the other mechanisms or the other regulatory mechanisms, right? Action potential. So the inside is negatively charged, outside is positive. For action potential generation, what happens? This flow of cations or positive charge from the outside to the inside so that the inside becomes positive. And this, once an action potential is generated, it promotes further generation of action potential. So it's a positive feedback cycle, right? And it's known as the Hodgkin's cycle. You need to remember. It's known as Hodgkin's cycle. Now, gastric acid secretion. Some of you might think that, it's an example of feed forward, or, why do we consider it as, uh, you know, so forgetting gastric acid secretion, let's focus on clotting. So in clotting, again, what happens? There's a cascade of 13 factors. Now, gastric acid secretion, I want you to focus. See, in the cephalic phase, it is that the sight of smell. So there's impulses from the medulla, which go via the vagus to the gastric glands, leads to secretion. In gastric phase, what happens? You have gastrin, which promotes the release of acid, right? So for gastric acid secretion, you have gastrin, which promotes HCl secretion. But now, when much more HCl is being produced, you cannot have perpetual production of HCl, right? So that is inhibited by something known as GIP. And GIP is gastrin inhibiting peptide or inhibitory in which way. So that's an example of negative feedback, right? So the cephalic phase is positive feedback, gastric phase is negative feedback. Even the intestinal phase, for example, when you have food or acid-rich food which reaches the duodenum, the pH decreases. The pH decreases, also there's release of somatos, there's release of, there's release of secretin and cholecystokine. So both of them, they inhibit gastric acid secretion, right? So I want you to remember that gastric secretion, the cephalic phase is feed forward, gastric intestinal. There are stimuli which support the release and then it is inhibited. So it's an example of feedback mechanism. Okay. Let's go to the next question. So the next question is, on infusion of a large volume of blood, the mean arterial pressure. Mean arterial pressure rises from 120 to 180 millimeters of mercury. Due to the baroreceptor feedback mechanism, the mean arterial pressure falls down back to 150 millimeters of mercury. Calculate the gain of a feedback system. Now, for example, baroreceptor, a very classical example of negative feedback. So what has happened in the question? You had a patient at 120, which is his mean arterial pressure. You have given him a lot of blood, and now the pressure has risen up to 180 mm of Hg. Again, after some time, the pressure comes back to 150 millimeters of mercury. So what do you see? That the initial value was 120. The deviation was up to 180. So the deviation was plus 60 millimeters. Now the correction was 30 millimeters. Yeah, so the deviation was 60. Correction post the deviation was 30. But how much is the error remaining? The error remaining is still 30. So what's the formula for gain? Gain equal to correction upon error. So the correction was 30. The error is still 30, right? So the gain is 1. In feedback, for example, any feedback system, you're trying to bring it back to normal. So the correction is in a direction which is opposite to the deviation. That's why you put in a negative charge. Or even by calculation, if you try to derive it, it would be, correction would be 180 minus 150. And what would be the error which remains? Error means original value minus the final value. So it would be 30 by minus 30. So it would be again minus 1. As I told you before, the minus sign indicates that the change is in a direction which is opposite to the direction of the deviation from norm, right? This another concept, for example, what happens if error is 0? So if error is 0, gain is infinity. And do we have a control system in our body which has infinite gain? That means it does not leave a trace of error. Yes, we have the renal regulation of blood volume, right? So kidneys maintain blood volume via the RAS mechanism, which is renin, angiotensin, aldosterone system, in such a way that the gain is infinity. So infinity means there's very little scope for error. Again, this is something which is heavily tested. Okay. So the next question. Now we are done with homeostatic mechanisms. We'll move forward to the cellular organs. What have we studied so far in homeostatic mechanisms? We have seen what homeostasis is broadly, which are the three control systems, their examples, and what is gain for a negative feedback system. You cannot have gain for positive feedback systems because there's no correction back to normal, right? It's only upwards, positive, right? So now let's go to something which is tested by the central institutes, which are very fond of asking questions on cell biology, which is an intense, uh, stream of research in itself. So the first question for cell biology is plasma membrane in eukaryotes is made up of all except. Now we know that the plasma membrane is made up of a phospholipid bilayer, right? So we have the hydrophilic, hydrophilic ends made up of the phosphate moiety, which is outside, and the inside we have the lipids, which are hydrophobic, right? So plasma membrane in eukaryotes, generally we say that it's like proteins which are floating as icebergs in a sea of lipids, right? So you have a lot of lipids, and in that you have proteins. Yeah, this is a 2D representation, 3D. It's like, yeah. So which are the lipids which you generally see in the plasma membrane of eukaryotes? You have cholesterol, yes. You have lecithin, yes. Do you have sphingomyelin, yes. Do you have triglycerides? No, you do not have triglycerides. Triglycerides are esters of fatty acid and glycerol. So triglycerides are generally absent. You have phospholipids, which is lecithin. You have sphingolipids, which is sphingomyelin, and you also have cholesterol, right? Cholesterol helps in maintaining the buffer of the cell membrane. That's why cholesterol is also known as the membrane buffer, okay? This is the buffering system. Right. So the buffering system of the cell membrane is the cholesterol because there'll be some fatty acids or some lipids which increase the rigidity of the membrane. There'll be some which increase the fluidity. So generally something which is fluid is better. In cell membranes, fatty acids which increase the fluidity are good fatty acids. Fatty acids which decrease the fluidity, not very good fatty acids. Cholesterol neither increases nor decreases. It acts as a buffer, right? So again, since it was asterisk, it was asked in AIMS once, and the answer was triglycerides. They are not found in eukaryotes. Okay. And who gave this model of cell membrane? That is a phospholipid bilayer. It's a bilayer. It was given by Singer and Nicholson. Right? Okay, let's move to the next question. Membrane fluidity is decreased by which type of fatty acid? So we were just discussing, right? So you might have seen ads which say that, PUFA or polyunsaturated fatty acids, good for heart, palm oil, which is bad for health because it is a saturated fatty acid. So we need to go one step above. We need to know which are the saturated fatty acids, which are the unsaturated fatty acids. So membrane fluidity is decreased by which acid do you think? So the answer is stearic acid. So fluidity increase by PUFA, which is unsaturated fatty acids. Examples, all the three: oleic acid, linoleic acid, and linolenic acid. All of them increase membrane fluidity. Which of them decrease fluidity? Which are saturated fatty acids. Your palmitic acid, main component of your palm oil. Then you have stearic acid. You have myristic acid. All of them, stearic acid, myristic acid, if I'm not wrong, they're generally found in soaps, used to prepare soaps. And how are soaps prepared? They're prepared by hydrogenation of fatty acids, right? So unsaturated fatty acid means the carbon either has a double bond or the carbon has a triple bond. And if you might have, if you remember your chemistries, in saturated fatty acids, the carbons are saturated with hydrogen. So when they are saturated with hydrogen, they have their configuration as sp3. It's a tetrahedral. It occupies, a lot of space, right? And there's very, for example, it's not a very compact thing. This one, sp2, sp. So this, if I'm not wrong, it's flat. Yeah, this is also linear. So they can accommodate much more. They can have more fluidity, right? Saturated fatty acids, they are very rigid. They cannot be accompanied well, right? So that's the difference. So membrane fluidity is decreased by all these saturated fatty acids. You need to know the names. You have palmitic, you have stearic, and you have myristic acid, right? Palmitic, stearic, myristic. And for unsaturated fatty acids, you have oleic acid, linoleic acid, and linolenic acid. Okay. The next question, which of the following is an example of peripheral proteins? So what do you think? The answer is cell surface receptors. Now, again, the wording is a bit confusing, but it's not that hard, right? So if you have your cell membrane, you'll have certain proteins which traverse the entire span of the membrane. These are known as integral proteins. They are traveling through and through, right? They can act as a channel. For example, this one. You can have movement of solutes. You can have movement of water, right? So that is an integral protein. Or you can have a carrier protein. Best example is the sodium potassium ATPase. Okay. So sodium potassium ATPase, if you see, you have again an alpha subunit, you have a beta subunit, right? So it takes in 3 sodium outside and it brings in 2 potassium. So yeah, 3 sodium out, 2 potassium in. You also have ATPase, which breaks down ATP into ADP and IP. This is ATPase, right? So you can see it can be a channel protein, it can be a carrier protein. You have something called as a G protein coupled receptors. Now, how are G protein coupled receptors, if you ask me? So you have, if this is your membrane, you have a receptor on one end, 5, 6, 7. Now, why did I count 7? Because those are known as serpentine receptors, or also known as heptahelical receptors. Again, they are traversing through the membrane. So anything which traverses through the membrane is an integral protein. So you have the sodium potassium ATPase pump, G protein coupled receptor, insulin receptor. Insulin is a very peculiar receptor. You have something which is known as a JAK-STAT receptor. Yeah. So JAK stands for Janus kinase. STAT stands for signal transduction and transformation, right? So the intracellular ends have tyrosine kinase activity. So whenever insulin binds, the receptor dimerizes. And then the tyrosine kinase is activated and the downstream process is done. Right? So again, insulin receptor also traverses through and through. So it's also an example of an integral protein. So cell surface receptors, unless otherwise specified, they are examples of peripheral proteins. Okay. Even G protein coupled receptor is a receptor, but it traverses through and through. If they have mentioned cell surface, obviously that becomes the answer because it does not traverse and then it's not an integral protein. Which of the following membranes has the highest protein per gram tissue? This was again an Ames question. So let's see the options. So you have myelin sheath, outer mitochondrial membrane, RBC membrane, inner mitochondrial membrane. So what do you think is the answer? So the answer is inner mitochondrial membrane. So myelin sheath, as the name myelin suggests, it has a lot of fat or fatty acid. So the fat to protein ratio is the lowest, right? It's 0.23. If you remember, it's okay. If you don't, also it's okay. Outer mitochondrial membrane, RBC membrane, both of them have a similar ratio, which is, so I should have written it as protein to, yeah, I should have written it as protein to fat ratio. So it has much more fat as compared to protein. That is your myelin sheath. Outer mitochondrial membrane, RBC membrane, the ratio is around 1.1. Okay. And inner mitochondrial membrane, if you remember the structure, the inner mitochondrial membrane is thrown into folds. And these folds are known as cristae. What's the main function? They have multiple complexes. Complexes 1 to 4 for cellular respiration. So it has a very rich array of enzymes and proteins, other structural proteins. Enzymes basically are, most of the enzymes are proteins again. So the inner mitochondrial membrane has a very high amount of protein present in it, right? So the clinical application or a pathological application is what happens in when the cell is starved, from where does it get energy? Generally, the inner mitochondrial membrane, which is very uh, rich in protein. So that is, what do you say? The lysosomes themselves eat those or fuse with those membrane bound, with those uh, organelles, and this is known as autophagy. Yeah, autophagy is the cell eating itself in times of starvation or nutrient deprivation. So where do you get maximum nutrients? You get it from the inner mitochondrial membrane, right? So in starvation, there is decreased use of energy. There is energy conservation. So you require less mitochondria. Okay. So long story short, inner mitochondrial membrane, highest amount of protein per gram of tissue. Myelin sheath, highest amount of fat per gram of tissue. Okay. Sequence of vesicular transport in a cell via Golgi apparatus is, again, this has been a past question. So what do you think is the answer? Yeah, so it is ER, cis, and trans and cell membrane. So if you have a nucleus, you have the associated rough endoplasmic reticulum. Rough endoplasmic reticulum, there is processing and packaging. The vesicles come out and then they are directed towards the Golgi apparatus. Now, Golgi apparatus is a series of tubules. Yeah, so you have the cis phase and you have the trans phase. So the cis phase is the receiving end. Trans is the releasing end. Cis is also known as the formation phase. So the vesicles go inside. They are modified by the Golgi and then they are released back. They are basically glycosylated or carbohydrate residues are attached, right? So carbohydrate residues are attached and then they are released and they go towards the cell membrane to be released outside the cell, right? So you have the endoplasmic reticulum, you have the Golgi apparatus, and you have the cell membrane. In Golgi apparatus, you have the cis phase, which is the receiving end. You have the trans phase, which is the releasing end. Okay. So again, this was a question. And who had discovered Golgi bodies? Golgi bodies were discovered by a scientist named as Camillo, Camillo, Camillo Golgi. Okay. All of the following are enzyme markers of cell membrane except. Now, again, it's a very hot question nowadays because it's difficult to memorize, but we'll try our best. Okay. So all of the following are enzyme markers of cell membrane except cell membrane is the organelle which is being asked. Okay. Sodium potassium ATPase pump. Yes, I just showed you previously that it's an integral protein or it's a carrier protein, right? 5' nucleotidase. Okay, we do not know. Adenyl cyclase. Yes, because your G protein coupled receptors, they use adenyl cyclase. Okay. So adenyl cyclase, yes. Glutamate dehydrogenase and 5' nucleotidase. Now, I know that glutamate dehydrogenase was a mitochondrial enzyme, right? So what remains? Nucleotidase. So nucleotidase has to be cell membrane again. So glutamate dehydrogenase is the answer. Now you need to know a few specific organelles which they are repeatedly asking, or it's good to know. For example, now cell membrane, you know that cell membrane is your sodium potassium ATPase pump, the nucleotidase, and the adenyl cyclase. All of them are for the cell membrane, which is there for mitochondria. So mitochondria, you have creatine kinase, right? Creatine phosphate gives energy. So its metabolism occurs in the mitochondria. You also have glutamate dehydrogenase. Okay. So creatine kinase, glutamate dehydrogenase for mitochondria. What is for lysosomes? See, lysosomes also known as suicide bags. They have all the enzymes for digestion. So you have cathepsin, you have acid phosphatase, right? Then you have something called as a peroxisome. So peroxisomes are generally related to free radical scavenging. Free radical scavenging, you require catalase, right? There's also an enzyme called uric acid oxidase. And peroxisomes, which you'll learn in biochemistry, is also responsible for oxidation of long chain and very long chain fatty acids. So that's for peroxisomes. What about Golgi apparatus I had mentioned before. It is responsible for giving carbohydrate residues. So that's why the carbohydrate is galactose. So the enzyme is galactosyl transferase. Okay. So Golgi bodies is galactosyl transferase. What about nucleus? Nucleus, you have your DNA and RNA polymerase. You also have an enzyme which is known as histone deacetylase, right? So histone deacetylase is responsible for epigenetic modifications. Again, it's a pathology thing. You can see one of my videos on cell injury. I've told you what epigenetics is. So epigenetics is changes in the, you know, heritable changes which are not caused by change in the base pairs or which are not caused by mutations. Those are epigenetic changes. So your sequence doesn't change, but your activity of transcription and translation changes. So that is epigenetics. It's above genetics. It's beyond genetics. Okay. For epigenetic modification, you have histone deacetylase, and it's an enzyme which is present in the nucleus. Okay. So let's move to the next. Okay, which of the following will test negative for all the mitochondrial markers? So which are the mitochondrial markers? Glutamate dehydrogenase and creatine kinase. Creatine kinase. Okay. So all mitochondrial markers, what's the answer? Obviously, it's mature RBC. Because RBCs don't have mitochondria. RBCs do not have a nucleus, do not have mitochondria. Okay. Spermatozoa, yes. Ova, yes. Zygote, yes. So the anatomy question is, zygote has mitochondria from which parent? It is always the mother, right? Because it's always the ova's mitochondria which survives. Sperm, only the head penetrates, whereas the neck piece was formed by the middle piece was formed by the mitochondria. That does not penetrate the zona pellucida, right? So all mitochondrial diseases are generally having which type of inheritance? It is maternal inheritance, okay? Let's go to the next question, which is the characteristic enzyme which is present in lysosome. So let's revise it again. Uric acid oxidase was peroxisome. Cathepsin, yeah, it was lysosome. Alkaline phosphatase. See, acid phosphatase was with lysosomes. Alkaline phosphatase is found in the endoplasmic reticulum, right? So you might have heard that which are the enzymes which are, produced by the liver, or in liver function tests, which enzymes do we see? We have SGOT, SGPT, and alkaline phosphatase. So alkaline phosphatase is there in the endoplasmic reticulum, which endoplasmic reticulum specifically smooth endoplasmic, because that's required for detoxification and other activities. Catalase was again peroxisome. So the answer is characteristic enzyme which is present is cathepsin. Let's go to the next question. Gap junctions are found in all except. Now, we have done homeostasis. We have done some important organelles. I'm sure that you might know much more about organelles than what has been discussed. Something which is not discussed frequently is the cell-to-cell junctions. And that's what we are going to discuss here. Okay. So it asks that gap junctions are found in all except neurons, cardiac muscles, smooth muscles, skeletal muscle. What do you think is the answer? Yes, the answer is skeletal muscle. Okay. So the answer is skeletal muscle. Why? Because as you know, skeletal muscle is a syncytium, right? It's an anatomical syncytium. Cardiac muscle is a physiological syncytium. Why is it called as a physiological syncytium? Because between two cardiac myocytes, you have free flow of ions, and that ions is because of gap junctions. So action potential spreads from one cell to other via these gap junctions. Okay. Smooth muscle, yes, smooth muscles also, they contract as a unit. That's why smooth muscle also has gap junctions. What about neurons? So you might say that, neurons, they are, they might be a synapse. Yeah. So between two neurons, the specialized junction is the synapse. You can have a chemical synapse or you can also have an electrical synapse. So if it is an electrical synapse, what will happen in electrical synapse? The presynaptic end is almost connected to the postsynaptic end, right? Via gap junctions. Okay. So even neurons. Skeletal muscle, what happens in skeletal muscle? Skeletal muscle itself, it is multinucleated. There is no cell boundaries here. Yeah, they were there earlier, but now they are not there in evolution. They've been lost. So it is like a continuous unit without any gap junctions. Okay. So gap junctions are found in all except skeletal muscle because it's an anatomical sensation. The boundaries are fused so that you have multinucleated. Each cell is multinucleated. Okay. Tight junctions are not present in cardiac muscle, small intestinal tubules, sertoli cells, choroid plexus. What do you think is the answer? As discussed earlier, cardiac muscle has gap junctions. So tight junctions are absent here. What about small intestinal tubules, Sertoli cells, and choroid plexus? See, whenever you have tight junctions means you want to form a barrier. So, for example, in the brain, you have the blood-brain barrier, which is formed by astrocytes, right? In the testis, you have the blood-testis barrier, which is formed by Sertoli cells. Choroid plexus. What is basically choroid plexus? Choroid plexus is a place where the CSF is being produced, and CSF is more often than not, it is derived from the blood vessels itself, right? But it doesn't want the other fluids or the CSF to go back into the blood vessels. So you have a barrier around again. So that is the choroid plexus. So blood-brain barrier is astrocytes. You have choroid plexus. Even Sertoli cells, they help in the formation of the blood-testis barrier. Small intestinal tubules, again, small intestinal tubules, if you see, they are lined close, the cells of the intestinal epithelium are close to each other. As a result, this pathway, the paracellular pathway, does not exist. You require specialized transporters to transport food from the lumen to the blood. So again, you have the intestinal blood barrier. Okay. So in small intestinal tubules, yes. Cardiac muscle? No, because cardiac muscle has gap junctions. Which of the following is not a cell adhesion molecule? Now see, you can have connections between cells. You can have connection between cell to the basement membrane. And you can have generally connection between cell basement membrane and the intracellular connection to the extracellular space. Intracellular space to the extracellular space. So generally cell to cell connection. Yeah, these are known as cell adhesion molecules, right? One of the most common, before solving this question, one of the most common cell adhesion molecule is desmosome. They are found generally in epithelial cells. Then you have some connection between cell and the basement membrane. So the cell is not being connected to another cell. That's why it is known as hemidesmosome. Okay. And then you have certain proteins which link the intracellular surface or the intracellular space to the extracellular space. We'll discuss that. So let's first focus on this question. Which of the following is not a cell adhesion molecule? Let's do this by elimination. Integrin, let's see, selectin, cadherin, unchirin. Now, if you know that your RBC cell membrane is made up of spectrin and unchirin, so it's a cytoskeletal protein. It is not a cell adhesion molecule. So the rest three of them are cell adhesion molecules. You have integrins, you have selectins, you have cadherins. Where do you generally find integrins? Integrins are generally found in tight junctions, right? Selectins, as the name suggests, you need to split it. They are again cell-to-cell molecules which have lectin binding domains. Okay. And cadherin is, again, the name suggests it is cadherin. So it is calcium dependent cell adhesion molecule. Selectins, you need to remember those have lectin binding domains or connection between cells. Cadherins, they have calcium, they are basically calcium dependent cell adhesion molecules. Integrins are in tight junctions. Unchirin, spectrin are in RBC cell membrane. Okay. So we'll discuss 2, 3 more questions. Okay. So identify the incorrectly matched pair of intercellular connections and their structural units. So what do you think is the answer? Yes, so the answer is hemidesmosomes. Why? Because if you have two cells, I've said that you have desmosomes connecting. Those desmosomes in turn are formed by desmoglein, desmocolin, and there are many other proteins. Okay. So you have desmoglein, you have desmocolin. Okay. Gap junctions, as I already mentioned, they are found in cardiac myocytes. So you have one cardiac cell which is connected to the other cardiac cell by a gap junction, and gap junction are connects connexon, right? So it is like this. It has 6 subunits, and each subunit is called a connexin, right? So gap junction is connexon. Zonula occludens is an example of tight junction. So tight junction, you need to occlude it, right? When I say the space is being occluded, that means there's nothing left because it's very tight. So you have claudin, cludin, and you also have integrin. Okay. Those are tight junctions. Okay. So desmosomes I've told you, gap junctions I've told you, zona occludens I've told you. Hemidesmosomes, again, look at this diagram. Hemidesmosome is connected here to the basement membrane. It's a very tight connection. And that's why it is integrins. It is not cadherin, right? Cadherin, it's a surgical correlation. There's a type of breast cancer called lobular carcinoma. So you have two cancer cells of the breast. They are connected by E-cadherin, which is a calcium-dependent adhesion molecule. This was in situ. In situ means it has still not crossed the basement membrane. It is still above the basement membrane. Now what happens if this connection is lost? E-cadherin connection is lost. Now these cells become mobile. They can penetrate the basement membrane, they can go to the blood vessel, and they can have distant metastasis, right? And the most common sites generally, it goes to the ovary, It goes to the stomach, right? So from the breast, it can go to the stomach, it can go to the ovaries because of loss of E-cadherin between those two cells. Okay. Integrin. Integrin connects with. So now, like I told you, you had a cell, you had filaments, you had the basement membrane, and you had the extracellular space. So your basement membrane is generally made up of collagen, right? And you have integrins here. This is integrin. So integrin is being attached above to uh, keratin filaments, intermediate filaments, and below it is being connected to something which is called as fibronectin, or there's one more protein called laminin. So you need to remember that collagen, laminin, fibronectin. How do they help? They help in attachment of the intracellular surface or intracellular space to the extracellular space. You have integrin here. Outside you have collagen, laminin, fibronectin. Inside you have keratin intermediate filaments. So keratin intermediate filaments are anchored collagen, fibronectin, laminin via integrins. So integrin connects with fibronectin. Okay. Force generating proteins, again, it's a very important question. Force generating proteins, which of them do you think they are? They are dynein and kinesin. Okay. So you should remember that there are three force generating proteins. There are dynein, kinesin, and myosin. Okay. All of these three are known as molecular motors. So for example, if you have your nerve cell and you require some things to be transferred across, so this is known as prodromic. This is the reverse transport. This is known as antidromic. So kinesin generally is prodromic. Antidromic is dynein. Okay. So many viruses, they travel in an antidromic direction. So that's one clinical, applied part of it. For example, rabies virus, polio virus, they have antidromic conduction. So kinesin, myosin, and dynein, all of them help in movement of substances along a particular path. That's why those are known as motors. What is motor? Motor is something which propels the car forward. So in this case, these molecular motors are propelling substances forward, which are synthesized by the cell, or in some cases, viruses as well. Okay. What is Cartagena syndrome? Now, we have done homeostasis, we have done feedback mechanisms, we have done cellular organelles, we have done cell proteins, and now we'll go towards some clinical aspects. So what is Cartagena syndrome? Cartagena syndrome is also known as primary ciliary dyskinesia. So your respiratory tract epithelium, it is formed by pseudostratified ciliated columnar epithelium. Okay. So how does this cilia act? Cilia, if this is your cell columnar, you have cilia. So this cilia always move in one direction. This is known as metachromism. Okay. So for example, if I'm breathing, the air is going in, my cilia are always beating upwards because they want all the dust particles, all the mucus which is formed, not to go into the lungs, but to go out. Okay. So now I'm having a cold, a pretty intense cold. So what is helping me from not letting many other pathogens or the mucus getting accumulated in my lungs, that is cilia, because cilia are constantly beating in the one direction. Okay. So that is known as metachromism. And these cilia, when you see the ultrastructure, when you see a cross section of cilia, you have something called as a 9+2 arrangement. So you have 9 spokes, and in the center, you have two more. Okay, so it's known as 9+2 arrangement, and these spokes are connected to each other. And these are made up of the protein responsible is dynein. So if there is some problem with dynein, what will happen? The cilia will not move. So if these cilia do not move, the secretions will keep getting accumulated. They'll keep getting infected. So if there's accumulation, pathogens will come and they'll infect it. They'll not be eliminated. So Cartagena syndrome is characterized by a triad of bronchiectasis, chronic sinusitis, and situs inversus. So the answer is all of the above. Bronchiectasis means what? You had bronchi. Now, ectasia meaning something which is tortuous. So you had bronchi. Normally, the cilia were helping in propelling the particles or the mucus forward. Now, in primary ciliary dyskinesia, so what would happen is the cilia won't act. So there's accumulation of mucus which will get infected, and as a result, the bronchi will become dilated and they'll become tortuous. That tortuosity is known as bronchiectasis. Okay. Chronic sinusitis, you have your maxillary sinuses, you have your ethmoid sinuses, you have posteriorly, you have your sphenoid, you also have your frontal sinuses. So all those sinuses, again, if you're repeatedly getting infection, what is the reason? Because your cilia are not moving well. Okay. So in Cartagena syndrome, there is chronic sinusitis. This sinusitis through and through. Okay, it's not occasional. So you have bronchiectasis, you have sinusitis, because again, sinuses are lined by ciliated epithelium, which pushes it forward. Situs inversus. During development, their cilia is also required for movement of organs in particular directions. For example, the heart is towards the left, the spleen is towards the left, the liver is to the right. So what determines it? During organogenesis or during development, there is cilia which is required for rotation of organs. So if there is malrotation of organs due to a defective dynein arm, what would happen? The organs would be swapped or reversed, right? So you'll have heart to the right, you'll have liver to the left, and that is known as situs inversus. Okay. So Cartagena syndrome is characterized by a triad of bronchiectasis, chronic sinusitis, and situs inversus. And the last question is, a patient with cancer was given paclitaxel. The drug acts on dividing cells by inhibiting mitotic spindles. These mitotic spindles are formed by. So you would have studied that in metaphase, during cell division, which are the 4 phases? You have a prophase, you have a metaphase, you have an anaphase, you have a telophase, right? So in metaphase, what happens is the chromosomes, they are arranged at the center, right? And from the two centrioles, you have tubules, which help in disjunction, followed by cell division. So what would happen is, if this disjunction doesn't happen, or if it is inhibited, the cells won't divide. So that is a property which we employ in cancer cells, right? Cancer cells are rapidly dividing. We don't want this division to occur. So this mitotic spindle is inhibited by drugs which are known as Vinca alkaloids. Vincristin, vinblastin, the very famous drugs. Paclitaxel, docetaxel, all of these drugs act by inhibiting the mitotic spindle formation. There's one more drug called colchicine. colchicine is used in gout again. Okay. Why gout? Because your neutrophils, 3 to 5 load, right? Your neutrophils go from the blood vessel to the site of gout or uric acid deposition. And that will lead to inflammation, more increase in inflammation. You don't want that inflammation. So this chemotaxis has to be inhibited. Again, for that chemotaxis inhibition, you use colchicine because the protein used is tubulin. Via tubulin, the cell moves forward. So colchicine is used in gout because it inhibits chemotaxis of neutrophils. It is, vincristine, vinblastine, they are used in cancers, example, breast cancer, because, you don't want rapid division. Right. So these mitotic spindles are formed by the protein, which is tubulin. See, keratin, vimentin, these are intermediate filament proteins. Keratin is found in all kinds of epithelium. Vimentin is formed with something which is mesenchymal. Okay. And ubiquitin is for protein degradation. So with this, we complete our discussion. I'm not sure if it was pretty helpful, but I did enjoy a lot. I think it will benefit you. And since it was the first time, I apologize if there were some technical issues or technical glitches. But do let me know in the comment sections below how did you find it, and if you liked it, do share it with your friends. And your constructive criticism, brickbats or bouquets, whatever it is, matters to me. Okay, so yeah, thank you.